My name is George Flynn, and this is my review of the Spectral Reflectance Imaging Biosensor. So what is a DNA microarray? A DNA microarray is a tool used to analyze and quantify DNA sequences that make up genes. It is similar to a computer chip and contains thousands of short, synthetic, single-stranded DNA sequences that together add up to make the normal gene in question, or a mutated form of said gene. This chip works by obtaining a DNA sample from a patient's blood and a control sample, aka a non-mutated sample. The next step is to denature this DNA into two complementary strands of DNA, cut them into smaller fragments, and then label each fragment by attaching a fluorescent dye. An example is labeling the patient samples with green while the control is labeled in red, as you can see in the image on the screen. After this, both samples are loaded onto the microarray chip and then combined to the synthetic DNA strands already located on the chip. If the patient has a mutation in their sample, then their fragments will bind to the mutated DNA on the chip, whereas the control will bind to the normal DNA sequences. Sequences shared between the patient and control samples appear as a mix of the colors, yellow in this case, while the other colors, aka red and green, will show up as their respective either non-mutated or control sample. This allows for detection of mutated DNA sequences in a patient, and as technologies advance, these chips have evolved to be able to include a large portion of the human genome. Alternatively, RNA can also be used with the microarrays to look at whether certain genes are turned on or off. This is extremely useful for clinical diagnosis and preventative treatments for those at risk of certain diseases. A new application for these DNA microarrays is to find single nucleotide polymorphisms, or SNPs, which are often indicators for developing potential diseases and treatment options. An important factor for SNP studies is the ability to resolve single mismatched hybrids from perfect hybrids through measuring different binding energies between the two. Different base pair interactions yield different binding energies, which allows for a method to detect where SNPs occur. With the conventional fluorescence-based detection methods, relating the microarray fluorescence intensity to binding energies is often difficult. Complicated procedures are required to accurately quantify genetic expression based on fluorescent detection, making reproducibility and reliability often questionable. The solution presented here is called the Spectral Reflectance Imaging Biosensor, or SRIB for short. This is a label-free detection technique, meaning that there is no need for a labeler such as a fluorescent dye. These kinds of detectors work through molecular mass, microcantilevers, acoustic waves, etc. In the SRIB, it works as a result of mass accumulation, which allows for the measurement of the change in optical thickness of a biolayer. It uses optical thickness to convert to mass accumulation through a conversion rate of 1 nanometer corresponding to a biomass density of 0.8 nanograms per millimeter squared. Doing this allows for them to quantify the mass change in the different samples. So they can convert from looking at an optical thickness to what the actual mass density of each sample is. This method provides quantitative information about the interactions between analytes and immobilized probes, allowing for detection of the hybridization of oligonucleotides and single mismatch discrimination. It also allows for a simple and accurate method for high-throughput oligonucleotide sensing and SNP studies, which, as defined earlier, is quite a problem with today's capabilities. So to test how well the SRIB performs, an array was used consisting of sequences containing perfect matches, single mismatches, or double mismatches. First, the SRIB scanned the sample before hybridization, then again after hybridization to measure the incremental changes in mass for each spot. The difference in grayscale intensity allows for quantification in the change of mass. In the experiment, the target oligonucleotide sequence was a 20 mer sequence, with the control being a 40 mer sequence. Different mutated sequences were hybridized to the sample to see how closely it matches the target sequence. A 20 mer duplex of the target was also introduced. Results show that all samples except for the 40 mer control and the 20 mer duplex of the target sequence show an increase in mass density. This is expected since there should be a mass change in sequences which differ from the target oligonucleotide strand. It could clearly distinguish between double-stranded mismatches. However, kinetic denaturation experiments were needed for single mismatches, and those were not as clearly seen. So since single mismatches were harder to quantify using the SRIB, denaturation experiments were done to better analyze this. First, an ionic concentration-dependent denaturation experiment was done to measure how each different sequence denatures compared to each other. This works because an ionic concentration of a buffer decreases, shielding the negatively charged DNA backbone is reduced. Which causes the shielding of the negatively charged DNA backbone to be reduced, decreasing stability of the duplex leading to denaturation. This denaturation is seen as a drop in mass density of the spots as seen in the first figure. As sol concentration is decreased, different sequences begin denaturing, allowing for different mutations to be distinguished. Next, the kinetics for each spot was determined as a ratio between each spot's mass density as it changed in time and its initial mass density. Blue shows the perfectly matched duplex, red shows single mismatch, and black represents a double mismatch. This kinetic quantification allows for an easy distinguishing between single and double mismatches, which previously were much harder to quantify. An exponential fit was also done to the decay, and a decay constant A2 was found and plotted for the 20-mer perfectly matched sequence, 18-mer perfectly matched sequence, the 18 and 20-mer single mismatches, and the 20-mer duplex for the perfectly matched sequence. This provides an additional visualization for easily distinguishing between different kinds of mismatches. With label detection such as fluorescent or the addition of mass tags to typically label-free mass spectrometry, there are many limitations that come with it. Limitations such as limited throughput in the case of mass spectrometry, or very high cost and bulkiness in higher throughput systems, and differences in surface functionalization, spotting, and labeling techniques that cause low reproducibility. An optical label-free detection method such as the SRIB system has been shown to bypass many of these limitations and allows for detection of hybridization reactions while also being very simple. Mass density comparisons in conjunction with the kinetics of denaturation has been shown here to allow for clear distinguishing between perfect and mismatched sequences. Overall, this method allows for a simple way to detect and analyze SNPs while also having the capabilities for high throughput, especially when compared to previous detection methods. Overall, in the future, I can see this technology as, well, maybe not being perfect now, having the capabilities for being improved and being really good for the field. Thank you for listening.